understand the mechanism of telomerase action, first of all we have to understand that what is telomere. Telomere is a repetitive sequence present at the end of eukaryotic chromosome. It protects the chromosomes from nucleolytic degradations, unusual recombinations, repair, and end-to-end chromosomal joining. Now, during replication, as we know, during replication, leading strand synthesize continuous DNA. But on the other hand, in case of lagging strand, the Okazaki fragments are formed, later RNA fibers are removed, and the gaps are sealed. So the RNA fiber which is present at the end of the eukaryotic chromosome in the lagging strand is not replaced, is not RNA primer at the end of the chromosome in the lagging strand is not replaced by DNA, because there is no vacant 3' OH that synthesize the DNA. So there is a gap. Is present at that region. So over multiple cell division or cell cycle, the length of the DNA which is present in the lagging strand decreases. So the length of the DNA decreases in the next replication process. So we have to protect the end. So it is a problem of, it is also called an end replication problem. Now the point, so to replenish the end replication problem, telomerase play a very important role. Now what is telomerase? Telomerase is a RNA dependent DNA polymerase, or it is a type of reverse transcriptase. So it is a reverse transcriptase that synthesizes DNA by using RNA as a template. So it is a type of ribonucleoprotein. So the telomerase is a type of ribonucleoprotein that add DNA and nucleotides at the 3' OH of the DNA. Now, so telomerase, what is telomerase? Telomerase is a reverse transcriptase. Telomerase is a reverse transcriptase because it adds nucleotides, on the 3' end of the DNA by using RNA as a template. So it contains RNA itself. Now telomerase is a type of ribonucleoprotein because it contains RNA and proteins. Telomerase generally absent in somatic cells. But it is active in cancer cell and gamete. So this is the 3' end of a DNA, and this is the after removing of the primer. As you can see from the Okazaki fragment, there is a gap region, and we have to maintain this region. So telomerase, this is the RNA of the telomerase enzyme. Telomerase content RNA. So this is the 3', 5'. This is the RNA sequences present inside the telomerase enzyme, and this is the telomerase enzyme. Now this telomerase enzyme uses its RNA as a template and add nucleotides to the 3' end. As you can see, TTH, so G-rich sequence. Telomere is a G-rich sequence like TTA, triple G, TTA, triple G, TTA, GGG, TTA, GGG. So as you can see, telomerase adds nucleotides at the 3' end. So 3' end is increases in length. Now in the third step, as you can see, so this is TTA, GGG, TTA, GGG. The telomerase adds nucleotide TTA, GGG, TTA, GGG. G. Now the primer adds and ultimately DNA polymerase synthesize the DNA. So DNA polymerase gets 3' OH and through which it synthesizes the DNA. And after the DNA ligase fill the gap. So as you can see, the length of the telomere is maintained. In case of 3' overhang is recognized as a damaged DNA. So there are several types of DNA repair mechanisms are active, several types of proteins are active that are involved in the DNA repairs, and ultimately cell cycle arrest and cell undergo apoptosis. So we have to protect the 3' end, 3' telomere region, you can also say. So how to protect? There are two important proteins, TRF1 and TRF2. These two important proteins binds with that particular DNA and form a T-loop structure to protect the DNA from degradation, nucleolytic degradations, DNA repair mechanisms, and other types of apoptosis mechanism. So if we inhibit the TRF2 protein, what happened? Inhibition of TRF2 leads to activation of ATM or P53 dependent pathway that ultimately arrests the cell cycle and cell undergo apoptosis. Now we have to protect the 3' overhang of the DNA. So as you can see, this is 3', 5', and the formation of the T-loop is initiated. This is the formation of the T-loop is initiated. So the first protein that is came TRF1. So TRF1 binds with that particular DNA, and after the function of the TRF1, TRF2 came and form the T-loop. So invasion of the 3' overhang occur due to the action of TRF2 protein. Inhibition of the TRF2 protein leads to activation of ATM or P53 dependent apoptosis pathway. Due to this, cell arrest, cell cycle arrest, and cell undergo apoptosis. We have to protect the 3' end because 3' end is recognized as damaged DNA. So this is the formation of the T-loop, and this is the D-loop structure.